Hello viewers, welcome once again to our ongoing series in genetics entitled Mendelism and Beyond. In my previous two interactions with you on the topic of sex determination mechanisms, I have presented before you a very detailed account of the chromosomal theory of sex determination. This of course included the X, Y and the Zs of the sex chromosomes met within different organisms. We also discussed at length the hormonal theory of sex determination and learnt about the fascinating marine worm Bonellia, where we found that all the larvae are cytogenetically identical, but their future differentiation depends on whether they would develop alone or whether they would be within the body of the female, and that is going to decide the fate of the sex. So in other words, here the hormones would either activate or inhibit a particular sex. I had also discussed with you the case of free marten or the hormonal intersex cattle. It was a very interesting situation where the free marten develops only when the fetal membrane of the two embryos fused together, such that cross circulation between the two embryos would take place. And during this cross circulation, some of the hormones which were released by male embryo would cross the placenta of the female embryo, and there it would inhibit the secondary sexual characters in the female embryo and at the same time induce male characters. As a result, the female embryo after birth would become more like a male but would be a sexually sterile female. This is what the free marten was. In fact, I had concluded my lecture on sex determining mechanisms part 4 by showing you a summary diagram, and today we are going to begin precisely from there on. The diagram on your screens again signifies that we have two types of factors: genotypic factors and the environmental factors. As you can see on the left hand side of your screens, the genotypic factors are reigning heavy than the environmental ones, and therefore the environmentally controlled sex determining mechanism is poor in this case, and we would have the normal distribution of XX, XY and XYZZ or the ZZZW type of sex determination. There would be other situations which we are going to discuss today, and they are the environmental factors, which could range from variations in temperature, salinity, photoperiod in case of plants, the pH, the density of the individuals, nutrition, various social factors and so on. In this case, the environmental factors would reign supreme regardless of the genotypic constitution of an individual. And in this case, we would find that the sex would be reversed depending upon how the conditions are prevailing. So, let us begin with the environmental theory of sex determination. Actually, this theory would state that the environmental factor plays a direct or an indirect role in determining the sex of an organism. As we know, every zygote will contain all the responsible genes for the development of sex of that individual. Expression of a particular sex whether it is a male or a female is going to be determined by the internal or the external environmental conditions. So what are these conditions? Under certain environmental conditions, we would find that a particular sex gene might get inhibited. So in that case, the other sex gene will get activated, giving only one type of sex. Alternatively, in other environmental condition, the process might get reversed and this would give rise to another type of sex. Let us take an example of a pteridophyte like equisetum. Now this is popularly called as puzzle grass. The spores of equisetum, when they are grown under optimum conditions, they will develop into a female gametophyte, and when grown under unfavorable conditions, they would develop into a male. On your screens, you would find a picture of the gametophyte of equisetum. Of course, you can observe the male sex organs and the enterozoids being released. So that means the direction of differentiation of sex in this case would be largely dependent on the type of conditions that are prevailing at that time. Other example we could take would be of cucumber and even melons. Here the daylight, that means the photo period, and temperature is also the driving force to differentiate the sex of a particular plant. A classical example of environmentally controlled sex determination pertains to the effect of temperature, wherein we would find that the classical examples are seen in case of reptiles. It is found that in some reptiles, they will use their incubation temperatures to determine the sex of an individual. So it would depend what is the temperature around the eggs, and that would depend as to in which direction the hatching would take place in the form of he or a she. So that means in some species it is found that if the eggs are extremely high or low temperature mediated, that means they are exposed to extremely high or low temperatures, they will become male. And if they are exposed to say medium temperatures, they would become female. So that means there are three different patterns, as you can see on your screens, which depict the temperature sex determination, as it is called, in case of reptiles. In some case, extremely high temperatures. In case we would find say very low temperatures, and in the other case the intermediate temperatures are going to decide as to what is going to be the fate of sex of the future individual. On your screens you can find that if a stack of eggs has been laid down, say for example by a turtle, it is buried in sand near the seashore, and all the eggs are not going to be exposed to the same temperature. The ones which are say exposed at the periphery to higher temperatures would develop into a male. The ones at the deepest point would again be exposed to a comparatively much lower temperatures, they would again be male. And the ones in the middle, which are say exposed to intermediate temperatures, they would develop into a female. So that means the whole chromosomal genetics is left alone, and it is the temperature which modulates the future sex of an individual. It was a scientist, Madeleine Charnier, who in 1966 first described the temperature dependent sex determination in a species of old world lizard, and that is the agama species, as you can see on your screens. Now, the case of turtles is very interesting because our children are born either males or females based on what their chromosomes are, whether they are XX or XY. I'm not talking about the aberrations. But then, unlike humans, as you can see on your screens, the sea turtles are not born male or female based on the chromosomes. They will follow a temperature dependent sex determination, which we popularly call as TSD. Now, this is different from the chromosomal sex determination. Now, that means it is only after fertilization that the sex of a sea turtle is determined, particularly at a stage, at a very specific stage, which we would like to call as the thermosensitive stage of embryonic development. So that means, what is the temperature of the nest? This is obviously going to influence the sex of an individual. If the nest in cooler sands is there, say if the temperature is below 27 degrees, then we would see more and more of male hatchlings. On your screens you would find a depiction of how the turtle's eggs are having different courses of embryonic development with reference to the rise or the lowering of temperatures. So that means the eggs which are incubated at much higher temperatures, say 30 degrees or above, they would produce more and more of female hatchlings. So that means if the temperature is fluctuating, sometimes it is very high and sometimes it is low. Now this is a confusing situation. This is likely to produce a mixture of male and female offsprings, because in this case, once the sex is determined and the hatchling is about to be there, then you cannot reverse it. It's a irreversible process. Actually, what happens is that we were talking about the thermosensitive period. On your screens you would find that actually the TSP or the thermosensitive period is so very vital. At this time, if the temperature is high, then this would result in a very high aromatase activity. Aromatase is an enzyme which is going to activate the female sex hormones, and as a result ovary is going to be produced. So we are going towards the femaleness, and if the temperature was low, so the activity aromatase is less, and as a result the ovaries are not produced, but instead testes are differentiated. So that means the temperature fluctuation itself is going to modulate the synthesis or the activity of aromatase enzyme. Now this case that we have just described is exactly reversed in a lizard by the name Tuatara. Here the males are going to be produced at higher temperature and the females would be born at low temperature. So that means both the type of situations are possible. For example, you will find in alligators, specifically the Mississippi alligators, there is a very specific range of temperature, say between 32 and 34 degrees Celsius, where males will develop, and below 32 and above 34 the females would come. So that means what is the impact of this type of a fluctuation? Obviously, if there are gross variations in temperature, it would definitely affect the sex ratio of species too. There was a scientist by the name Sharp. As early as in 1934, he also talked about the nutritional theory. He was of the opinion that the control of sex is not merely genetic, but then how the gametes are nourished. and he was of the opinion that if there is a good amount of nourishment, ample nourishment, then the direction is towards femaleness, and otherwise it would be the male ones. Let us take a case of another marine worm, which is Dinophilus. Here the size of the egg is going to determine the nature of sex. If the egg is of a small stature, it would definitely give rise to male progeny. Whereas if the egg is, say, about 27 times larger than the average one, and then it would have lots and lots of nutrition to look after the next generation, and then it would develop into a female. Sex of certain organisms is taking a particular direction which is going to affect the sex ratio, even as a result of infection by certain bacteria. Now, this is a very interesting case. As you can see on your screens, some of the insects, for example, the arthropods, the sex ratio is affected by infection of the bacterium Wolbachia. Now, it's a very interesting bacterium, because some of these species, even though they may be having chromosomal sex determination, but then once this bacterium infects them, the males are killed or they are feminized. On your screens you can see an electron micrograph of Wolbachia. This is within an insect cell, and it is infecting and changing the sex of an individual with the help of its secretions. Viewers, I must emphasize here that Wolbachia is a very potent antiviral bacterium, and therefore it holds a very great promise to control viruses like Zika and dengue, which are in the form of worldwide dangers to our population. And people are also talking about its potential in controlling malaria. Actually, what happens is the infected females can pass the bacterium on to their offspring in their eggs, and therefore the maternally inherited endosymbiont will automatically infect the next generation. Wolbachia in fact can spread very rapidly through a population by changing the reproductive potential of its host. And in this case, the infected individuals will definitely be on a reproductive advantage. You must remember, viewers, that this is a case of cytoplasmic incompatibility, where only member of a particular sex is able to reproduce, and therefore the uninfected females which are mating with the Wolbachia infected males, they are not going to produce any progeny, whereas there is no reduction in the number of offspring infected females produced. So no matter whether they mate with infected or uninfected males. So that means the infected females have a very strong selective advantage, and this also changes the sex ratio of a particular population. Another good example of the sex determination catalyzed by the environment is of a nematode, Gasteromyrmex. Now, this particular nematode parasitizes the aquatic larvae of a mayfly, that is Batis species. The females become sterile, and some of the males which are parasitized, they will become either intersex or they will become completely female. However, all the parasitized males and the females, they will behave like females in their future course of life, that is in their adulthood, and they will behave like females because they will crawl into the water as if to lay eggs, and this will help the parasite to return and infect the next generation. This again affects the sex ratio of the population. Let us look at the screens for another classical example of a marine annelid by the name of Ophryotrocha species, which exists in the northeast Atlantic shores. Now, this particular annelid has the capability to change its sex with the age of an organism. So, while it is young, it is a perfect sperm producing male. As it becomes older, it now transforms itself into an egg laying female. It is very interesting to know that if the parts of the older female are amputated surgically, it again assumes a male form. So that means here the sex determination seems to be related to age. Another classical example on your screens is of a sea anemone, Metridium senile. Now, as it grows, it changes its sex, and initially it was male. As it becomes larger, it becomes a female. So that means why do we need to have a larger size females? Obviously, the bigger size females are perhaps needed to produce more gametes for reproduction. There is another very important aspect of sex determination through the environmental control, and that is exemplified in a wrasse which is called as the thalassoma. Now, this is a native of the coral reefs of the tropical waters of the western Atlantic Ocean, and this is actually a fish, and it is a protogynous sequential hermaphrodite. Let us break these words. Protogynous means female initially. Sequential hermaphrodite may change into something different a little later. So all of them are born females, and some change the sex to male after maturation. So this dominant fish in the population would become male, and the remaining ones, they would be females. So the males would reach an initial phase when they can breed in groups with females, and some males will grow even larger and reach full coloration. So this is called as the terminal phase, and these large males will aggressively chase away these smaller ones and seek females to breed. And how would we know that they are ready to mate? The state of readiness is indicated by the different colors which they change in. Another very important example is that of a clownfish. The tropical clownfish, as you can see on your screens, the dominant individual in a group becomes female, while the other ones, they remain as the female. So that means the fish would live in groups, the dominant female would be there, and there would be smaller males. If you remove the female, the largest male becomes female. So that means again here the enzyme aromatase is the one which controls the androgen-estrogen ratio, thereby controlling sex. Another classical example of sequential hermaphroditism is seen in the mollusk Crepidula. This is also called as slipper limpet. Actually, each individual in this case also, this is a marine mollusk, it can be both male and a female, but not at the same time. All the genes for male and female reproductive systems are present. The young mollusk is a functional male. It also has sperms, but then it doesn't have any ova. When it matures it transforms into a functional female. The genes activate at different time periods of the life cycle. On your screens, viewers, you can see the slipper limpet, which is present in the shallow sublittoral coastal areas. As the larva settles, now alone if it is there, it will now transform into a female. Other larvae will be attracted and they will fall on top of this female larva and they will become males and mate with it. Now with the passage of time, these males will become females. They will attract in turn more larvae. So that means they will settle one on top of the other. So all the lower ones would be transformed into females and only one which is at the top would be the male one. The other example is of a parasite Seculina. It goes on the crabs, and these particular crabs also change their feeding habits, and the parasites, they reproduce on themselves. Then in addition, there are various social factors. One should remember that there are certain fishes which may begin in a coral reef as of a particular sex. If the dominant male or female is gone, then it will be a triggering factor for the change in sex of the remaining ones. So that means we would find that many a times sex determination is not only through the sex chromosomes, the environmental factors also play a very, very important role in this type of a situation. Sometimes even competition in a community may lead to sex determination, and some species can actively change the sex ratio of the offspring. So just to conclude, in many cases sex may be determined by various extrinsic factors after fertilization. It is not necessary that all these sex inducing or sex determining mechanisms are purely based on chromosomes. So we end our discussion again with the slide that we began with, that in certain cases it is the environmental factors which outweigh the genotypic factors. Thank you.